Hello, everybody, and welcome back. Today, we're going to be talking about Hardy-Weinberg equilibrium. And this is a picture of one of my favorite things. This is a lungfish. A lungfish is it's a fish, but as the name suggests, it can breathe air. And it's thought to represent kind of the lineage that eventually led to the colonization of land by vertebrates. So that's why I've always found this animal very fascinating. Now, in the early 2000s, we completed the human genome, and we found that humans have a genome that's about 3.2 billion base pairs in length, meaning that there's 3.2 billion nucleotides that make up the entire genetic code of human beings. And of course, in our arrogance, we thought that it must be the biggest genetic code that's out there to explain how wonderful we are, right? But when they started doing genome projects on all different sorts of species, we found that even something like the grain of rice can have many, many more times the base pairs that we have, suggesting that could be way much more genetic information. In the case of the lungfish, it has a genome that's about 133 billion base pairs, right? So that's 130 billion base pairs more than us, right? So what might be the deal here? Why might this be the case? Why aren't humans would have the largest genetic code to explain all of those genes that make us so wonderful and able to do all these things like, invent iPhones and shit like that? What's going on? Well, this is a measure of base pairs, so DNA sequences and how those nucleotides line up. But the reality is that most of the genome is what we call non-coding, meaning that it's DNA that it does not necessarily code for a gene or any sort of product, and instead is just there, right? In the case of humans, it's estimated that about 98% plus of the DNA sequences that are there are non-coding, with the rest being actual functional genes that can then produce a protein through transcription and translation. And it's thought that this non-coding DNA, since it's not producing anything, must be free of natural selection. Now, that's a useful tool in and of itself because we can essentially trace it through time, and we can look at relationships between species and between people, right? If we want to assign paternity or something like that. And we can also use it to develop biomarkers for particular diseases. But the assumption is that they're free of any natural selection. So, how might we know that that's the case? That's the big question. Well, the answer to that question is quite complex, but basically, you need to start somewhere with some kind of null hypothesis or a model that takes into account the fact that nothing is impacting this area of the DNA that you're interested in. So we talked before about the forces of evolution, right? Mutation, gene flow, non-random mating, genetic drift, natural selection, all of these operate as forces of evolution, which we define as changes in allele frequencies in a population over time. And if you have enough change and enough time, you can end up with some pretty big evolutionary changes, right? So you need a null model where this sort of thing does not apply, right? A null model where there is no selection, there is no genetic drift, the population is so large it's unaffected by gene flow, and that mating is completely random, right? And mutations aren't happening. So let's talk a little bit about what all this means, right? So here's a funny little, this is a little dated, but I remember these commercials. I thought they were hilarious. Most interesting guy in the world. So let's answer some of these questions. What's a population? Well, I would define a population as being all of the individuals in a given geographic area that have the opportunity to interact and mate with each other, right? So because of that, they're in the same population. If you have another group of the same species that happens to be on the other side of a mountain range and you can't get to them, then they would be considered a different population, right? Now within that population, you have a gene pool. So a gene pool is basically the population of alleles that are present there, right? So every individual is going to have their own suite of genes, their own suite of alleles for those genes. So we take the whole area into account. That's the gene pool, right? Now, what's the difference between genes and alleles? Well, if you recall from earlier lectures, an allele is just a version of a gene. A gene is coding for a specific protein. It's a specific area of the DNA, right? It's a specific site, a specific set of base pairs, which we call the locus of the DNA. But you can get variation, how many base pairs, which base pairs are present, and that's going to affect how that protein's going to look. And those subtle differences at that gene are what we call the alleles, right? So humans have the same genes, but we each have different combinations of alleles for all those genes, right? So a gene would be having a car. Allele would be what kind of car do you have? Do you have a sports car? Do you have a van? Do you have a truck? Right? So it's a version of the car that you have. And again, evolution. Evolution is just the changes in the frequencies of alleles in a population. That's at its simplest form what is happening. So what sort of things might cause alleles not to change? Well, allele frequencies are going to change if there's some force of evolution acting on it. If there is no force of evolution acting on it, then we don't expect those allele frequencies to change, because why would they? Right? So you can remove genetic drift by having a very large population size so that little variations in size here and there don't matter. You could have a population that's completely isolated, so there's no gene flow, no new genes coming into your population, none of your genes in your population leaving, right? No mutation, no new alleles springing up out of nowhere, right? No sexual selection, meaning that any kind of mating for the next generation is going to be 100% random, right? So there's no tendency to select the biggest or the baddest or the toughest or the most colorful or whatever it is, right? So mating is 100% random, like those first few years of college. And then no natural selection. Everyone is as fit as can be. Everyone's going to survive. No individuals are going to be more fit than another. Everybody makes it. So I think of this as, Disney's Zootopia. And if you've ever watched Zootopia, it's the story of a big city where all the animals live in harmony. Lions are walking down the street with wildebeest, foxes are hanging out with rabbits, and, sloths are driving cars that are way too fast for them, and they're working at the DMV, right? So it's an environment that doesn't seem to have any agent of selection happening there, right? There's no natural selection, and there's nothing like that, right? So perfect Zootopia. So under those conditions, you wouldn't expect allele frequencies to change. So thus evolution wouldn't happen. So in a population that's not evolving, what would we expect of allele frequencies? Well, simple. If you have allele frequencies in a population and nothing is acting on them, then you would expect them not to really change much. So we talk about this sort of situation as a population at Hardy-Weinberg equilibrium. Now in nature, as you can imagine, this is very rarely the case. But what the Hardy-Weinberg equilibrium does, and this is where a lot of people get confused, is it serves as a null hypothesis. It serves as a model of what the allele frequencies would look like if nothing is happening at all. And we use it to compare it to real data from real populations and natural populations. So it serves as a model by which we can compare and assess genetic changes happening in the wild. So in order to understand Hardy-Weinberg equilibrium, there are two equations. These are important equations. If you're interested in biology, you need to just memorize them. If you have a hard time memorizing them, just get them tattooed on your body somewhere. I would do it backwards so that you can read it when you're looking in the mirror. The first is the equation for allele frequency. And allele frequency is basically you minding your P's and Q's. P represents the frequency of the dominant allele, Q represents the frequency of the recessive allele, and these two added together equal 1. So what that tells you is that P and Q, being a frequency, is going to be a number smaller than 1, right? It's going to be a decimal. So it could be 0.5 plus 0.5. It could be 0.2 plus 0.8. It could be any of those things. But they add up to 1 at a Hardy-Weinberg equilibrium. So that is your allele frequency in a two-allele system. Now, to assess genotype frequency, so here we're talking about homozygotes. Homozygote dominant would be PQ, it would be P squared. Homozygous recessive would be Q squared. And 2PQ would represent your heterozygote condition. Again, these are frequencies, so the numbers will be less than 1, and they will add up, all three of them, to produce 1. So 100% of your alleles in a two-allele system. So again, you have a separate equation for allele frequency and a separate equation for genotype frequency. Once you have one figured out, you can figure out the other, and you will find that you're using these two interchangeably. And you kind of need to use them interchangeably in order to answer real questions. So let's take a system here. Here are some cats. You have black cats, you have white cats. The black cats and the white cats are determined by two alleles. A dominant allele B, which represents your P, a recessive lowercase b, which represents your Q, right? So you have your frequency of your dominant allele being P, your frequency of your recessive allele being Q. All of these frequencies add up to 100% in a population, i.e. you're accounting for everybody. So we know that P plus Q equals 1. Now, you also know that because it's a two-allele system, you could have homozygotes for both the dominant and the recessive, as well as a heterozygote that has both alleles. So when you're counting the alleles, you use the P plus Q equals 1 equation. But when you're counting individuals or genotypes, that's when you use the other equation, right? The frequency of homozygous dominant are basically your P times P squared. Homozygous recessive is Q times Q squared. And then the frequency of your heterozygotes is going to be 2PQ, right? And all of them are going to add up to 100%, i.e. 1, right? So depending on the question, this is important. When you're answering a question regarding Hardy-Weinberg equilibrium, you've got to make sure you understand if they're asking for the alleles or if they're asking for the individuals or the genotypes, because that'll determine how you basically answer the question, right? Even though in the process you'll figure out most of the details, just because you have to. So again, when we talk about counting alleles, we're talking about the capital B and the lowercase b. When we talk about counting individuals, we're talking about the genotypes. All right, so this is just showing the same thing again. So let's use our Hardy-Weinberg equilibrium. We have a population of a convenient number, highly convenient, 100 cats. We're going to assume that this population is at Hardy-Weinberg equilibrium. And you go in this population, you find that 84 of these cats are black. 16 of these cats are white. So how many are there of each genotype? Well, the easiest thing to do is to find the recessive phenotype, because you know that if you have a white cat, it can only be the homozygote recessive, right? Whereas if you look at the black cats, it could be a dominant homozygote or it could be a heterozygote. So you don't quite know which one it could be. So always try to figure out the homozygote recessive condition first, right? Or the rarer condition first. So in this case, we have 16 white cats. Well, conveniently, it's 100 cats in total. So the Q squared, we know that it's basically 16% of those cats are going to be the white cats. Another way to express 16% is just to say that Q squared equals 0.16. Right? So right off the bat, 0.16 Q squared. So now what? Well, now we have Q squared equals 0.16. We could solve for Q. We know that Q squared equals 0.16. The square root of that would be 0.4, right? Because 0.4 times 0.4 would be 0.16, right? So now we have the genotype, or now we have the allele frequency for Q. Q equals 0.4. And we know that P plus Q equals 1. So because we know Q is 0.4, we know that P is going to be 0.6. So 0.6 plus 0.4 equals 1. So 60% of the alleles are going to be P, the capital B allele, and 40% are going to be the Q. Well, now that we have that information, it's just a matter of solving it. So 0.6 times 0.6, that'll give us our homozygote dominant, our P squared. 0.6 times 0.6 will give us 0.36. And we know that 0.36 is our homozygote dominant genotype frequency. 0.16 is our homozygote recessive frequency. Now if we do 2 times P times Q, so it's 2 times 0.6 times 0.4, we end up with 0.48. So about 48% of these cats are going to be heterozygotes, 16% are going to be homozygote recessive, and 36% are going to be homozygote dominant. So there's your information right there. That can now serve as your null hypothesis. Again, P squared equals 0.36 in this population, 2PQ equals 0.48 in this population, and Q squared is equal to 0.16 in this population. That's our model, our null hypothesis of Hardy-Weinberg equilibrium. Now suppose that's what you expected, but you went out into the field and you caught the 100 cats and you actually genotyped them because you just happen to have the money and time to do that. And you actually find these numbers instead. P squared is 0.74, 2PQ is 0.10, and Q squared is 0.16. These are the data that you've sampled from the wild. So what might be an explanation for this data? Well, again, you're looking at your comparison with the Hardy-Weinberg equilibrium, your null model. So right off the bat, you notice that Q squared hasn't changed, right? So there doesn't seem to be anything going on with the homozygote recessive, but you do notice that there's a big disparity in the heterozygotes between what's under Hardy-Weinberg equilibrium and what's being sampled in the wild. And on the flip side, you have less heterozygotes in the sampled condition, and you have more homozygote dominant in the sampled condition. And then you do it at Hardy-Weinberg equilibrium. Again, Hardy-Weinberg equilibrium suggests that there's no force of evolution working here. And so you see this data and it tells you, wait a minute, there seems to be something acting here. It seems to be something maybe, giving the heterozygotes a disadvantage, right? Or maybe it's just better to be a homozygote dominant as opposed to a heterozygote, right? Maybe something about that heterozygote condition is a disadvantage, right? Or something could be favoring being a homozygote dominant. We don't know what that is. Either way, it doesn't seem like anything particular is acting on the homozygote recessive condition. So perhaps one of those conditions of Hardy-Weinberg equilibrium, one of those forces of evolution, is being violated as a result. So this is an example where you're comparing it and it allows you to make some guesses about, okay, what sort of things might be going on in terms of selection, right, in your population. So again, Hardy-Weinberg is a null model that allows you to compare. So if you have allele frequencies that don't seem to change between the parent and the offspring, then you could make the assumption that no evolution seems to be occurring. And this is something that's been tested and verified in the lab. It's been tested and verified in the field as well. And it serves as a wonderful way to study, especially small populations that are at risk of losing genetic diversity. So I have here a picture of a cheetah. Cheetahs have highly fragmented populations, and there's always concerns about their genetic diversity being very, very low, right? So what you find is these cats often have low genetic diversity and alleles that don't change through time, right? Because there doesn't seem to be selection acting because they're such small populations and things like this, right, in the lab or captivity. So there's a big emphasis on moving them around. So it allows them to assess genetic health in these populations. Okay, so let's do another example now. You have roses. You have white ones, you have red ones. Red ones are associated with the R allele, W associates with the white roses. And you have a population of 500 individuals, so there's 500 roses. 320 of those roses are going to be homozygous dominant, RR, which equates to 64%. 20 of those roses are going to be homozygous recessive, WW, and that equates to 4%. And then 160 of them are going to be heterozygotes, which equates to 32%. So the question we're going to ask is, what is the frequency of the R allele, and how many total R alleles are there in this population? So here it's slightly different. We're giving you the number of individuals, we're giving you the percentage, and it's up to you to find the frequencies of the alleles, right? Now, right off the bat, if you're given percentages, you can turn those into decimals very easily, right? So 64% would be 0.64, 4% would be 0.04, and 32% would be 0.32, right? So you could answer this most easily with a simple Punnett square, right? So if you take 0.64 and you find the square root of that, right, to give you your p squared, and you find the square root of that to give you your p or your allele, you'll find that it's 0.8. If your R allele is 0.8 or 80%, that means the remaining 20% or 0.2 is going to be the allele frequency for the W allele. Now, once you have that, it's just a matter of doing the multiplication. So you can see it all right here, right? You take this Punnett square, you have the RW, you could figure out all of the different variants that you need to figure out, right? All the different genotypes. So when you do this, in the next generation, let's say you take a heterozygote and you cross it with another heterozygote, and you do this again, you'll find that there's no difference. You still have this 80-20 split between the red and white allele, and they haven't changed. So why haven't they changed? Well, it's probably because they're in Hardy-Weinberg equilibrium, right? So even though you've bred them another generation, it's still. So one thing about genotypes, and especially allele frequencies, is sometimes the recessive allele frequencies can be recessive alleles can be very deleterious, and often they're expressed in their homozygote recessive form. So because you have a lot of these recessive alleles and they can be deleterious, so for example, a homozygote recessive condition can lead to something like polychoria here, where a person in one eyeball has two irises, which is a genetic condition. And a lot of diseases are this way. Why do they continue to persist in a population? Especially considering that selection would act very heavily on this. Well, the reason is simple. Heterozygotes. Alleles are going to hide in the heterozygote. In the heterozygote, you have the dominant condition, which could be something that's favorable, but every now and then you're going to get the recessive alleles surfacing up. And in a population, especially with inbreeding, you'll start getting those recessive alleles after becoming more and more common. And as that happens, that could lead to a lot of these otherwise fairly rare conditions and diseases to become more prevalent in a population. But those heterozygotes can essentially hide recessive alleles that can be damaging. So this sort of stuff very much has implications for a lot of things. And the Hardy-Weinberg matters, not only from a medical context like this, where you can use it to figure out what are the likelihood that alleles are going to show up, but also from a conservation perspective. This is a bird that I really like. It's called the Regent Honeyeater. It's a bird found in Australia. And recently it made the news because it's a bird that is so rare that the offspring have not heard adults sing. So it's a bird that has essentially forgotten its own song. And they need their songs in order to reproduce. So you've been seeing these declines in populations of them, and they're very rare now. And they've lost their ability to sing, right? So the captive breeding programs are there, and they're trying to instigate a captive breeding program right now. But they need to make sure that they get a wide variety of genetic diversity in order to prevent things like inbreeding depression, which happens when you have rare alleles that are deleterious showing up over and over and becoming more common in a population. You want to have a healthy, genetically diverse population. And this is a big problem with captive animals, right? Yeah, he's on there, Tiger King. Um, you know, the genetic integrity of species, especially species in captivity, which often have the goal of being released into the wild or to preserve animals that are otherwise rare or plants that are otherwise rare. We need to understand how their genetics play out. So that's where Hardy-Weinberg comes in. It allows us to essentially assess allele frequencies. Are they diverse enough? Are they, what prevalent, which genotypes tend to be prevalent? Are we overrepresenting some over others, right? And of course, the rarer the creatures, the more of a problem it is, right? So animals in captivity are often an issue. And many times, Hardy-Weinberg can reveal situations of very low genetic diversity because they deviate from Hardy-Weinberg, right? Because they violate those conditions of having a large population, right? And things like that. So the bottom line is this stuff's very important, a lot more important than Tiger King is. Um, I want you all to enjoy yourselves, learn a little bit more about Hardy-Weinberg. Once you do it a few times, it tends to be pretty straightforward. So I would encourage you to go out and find as many questions as possible. And with that, I'll leave you to it until next time. Have a good one.